We're moving on to the last lecture of this session, which will be given by Saurja DasGupta from together with Jack Szostak from Harvard University, and the title of his talk will be Prebiotic RNA Assembly, Chemistry, Catalysis, and Compartmentalization. The most difficult word in the original. Okay, please.
Hi. Thanks, Conan, for the introduction, and thanks to Amit for reaching out to me earlier when the meeting was supposedly going to be held at Israel. I was hoping to be there, but hopefully next year. So all of us trying to unravel the mysteries of the origins of life, we kind of operate within this thin, dark slice of unknown that is sandwiched between the worlds of known chemistry and known biology. And we're desperately trying to bridge this gap by shining light on different areas within this darkness, which is why sometimes we end up with slightly different answers to some of the pressing questions in the field. But looking at life as we know it, I think the worlds of chemistry and biology would have been bridged by a primitive cellular system generated from the self-assembly of non-living matter that would have at some point exhibited the emergent properties of life. So such a primitive cellular system, or a protocell as we like to call it, would minimally consist of genes, enzymes, and some sort of a compartment to put these molecules in. And the situation is further simplified by the fact that both enzymatic and genetic functions can be embodied by a single molecular entity, which was introduced by Sandeep. So thanks to Sandeep. This is RNA. RNA can act as both enzymes and genes. So considering the central role of RNA in the origin and evolution of early life, I'm trying to develop experimental models to demonstrate the various major chemical transitions that RNA must have gone through before the emergence of RNA-based primitive cellular life. So I'm going to briefly touch upon the different ways in which I'm doing this. But before all of that, I'd like to give you a very quick overview of what sort of transitions I'm talking about. Okay, so we all know that we started from this messy collection of chemicals, and then somehow we ended up with this vast diversity of well-organized life. So how did this happen? Of course, prebiotic chemistry generated the building blocks of RNA or RNA-like genetic polymers. And then those building blocks had to be chemically activated so that they could come together to form longer and longer oligomers. So our group about 4 to 5 years ago discovered 2-aminoimidazole as a very useful activating group in this regard. And interestingly, this molecule is also formed as a byproduct in the nucleoside synthesis pathways in prebiotic chemistry, which means that this molecule is prebiotically relevant, which is always a good thing in our field. So as these activated building blocks slowly come together and form longer and longer oligomeric chains, these oligomeric RNAs would then fold up in 3 dimensions and adopt complex, interesting structures, and some of these structures would likely show some catalytic function, which means they would behave as RNA enzymes or ribozymes. So this transition from non-enzymes to enzymes, I think, would have been a monumental step toward the origin of life. But there are, I think, 3 innovations that would have been really important for this transition to happen smoothly. So the first one is the emergence of ribozymes that would take the building blocks of non-enzymatic assembly and then use them as substrates for enzymatic assembly. The second one is that these ribozymes had to function under the conditions of prebiotic chemical activation of these substrates. And then third, of course, is a very logical transition, which is that the very first ribozymes had to have come from the self-assembly of self-assembly of activated building blocks through non-enzymatic assembly, right? Because the first enzymes had to come from non-enzymes somewhere. The fourth transition that I'll talk about is somewhat different but related. So we know that the modern RNA building blocks, NTPs, are triphosphate activated. But the problem with triphosphate activated building blocks is that they're really unreactive for non-enzymatic assembly, which is why we need these more intrinsically reactive building blocks, these imidazolide building blocks. So then at some point in RNA's journey, these ribozymes had to evolve the capacity to make longer RNAs from triphosphate activated building blocks as well. And the final transition is of profound importance, of course, is the emergence of compartmentalized catalysis, where ribozymes would function inside these prebiotic compartments and catalyze metabolism, trigger growth and division, and perhaps even invent Darwinian evolution. So let's start at the beginning. So I'm trying to connect non-enzymatic RNA assembly with enzymatic RNA assembly, and I'm going to use RNA ligation as a proxy for RNA assembly because it's easier to study. So the task here is to identify ribozymes that would use amino-imidazole activated oligomers as their substrates for catalytic ligation. So the way to do this is through in vitro selection, where I start with this diverse collection of RNA molecules, excuse me, and then I want to select sequences that would catalyze the ligation of 2 pieces of RNA. The first piece, called substrate 1 here, is connected to each of the sequences in the library through a linker, and the second piece is supplied in the reaction. This piece is 5' imidazole activated, and it also has a 3' biotin, which we use for the selection purposes. So this is how it works. Only those sequences in that library that catalyze this ligation reaction would get joined with the biotin tagged substrate, which means that these sequences would themselves get modified with the biotin, which means that we can pull these sequences from the inactive sequences using streptavidin capture, and these sequences would be pushed forward in the selection. And by repeating this cycle several times, we were able to identify several distinct classes of these ligase ribozymes. So the first generation of these ribozymes catalyzed the ligation of 16 nucleotide activated RNA oligomers, but we found that these ribozymes didn't do that well with shorter activated RNA substrates, which is why we carried out further rounds of evolution and selection to force these enzymes to join shorter and shorter substrates. And the most recent generation of these ligase ribozymes can work with activated substrates that are as short as 4 nucleotides. So I think some of you might have realized where I'm trying to go with this. I'm trying to evolve these ligase ribozymes into polymerase ribozymes that use 2 amino-imidazole activated monomers as building blocks. So the next obvious question is that, these ribozymes need activated substrates. Where do these activated substrates come from, especially on early Earth? So John Sutherland's group in the UK has suggested a very interesting chemistry where they've used amino-imidazole, isocyanide, and aldehydes to activate nucleoside monophosphates, so monomers. So we thought that we could use the same chemistry to activate our oligomeric substrates. And we also wanted to see if our ligase ribozymes were active in the presence of these chemicals. So before we went into ligation, we wanted to make sure that we optimize our activation chemistry because, we want to do everything in one pot, which means that if there are still a lot of unactivated substrates swimming around in that pot, they would bind to the template and inhibit ligation. So we optimized each component in this activation chemistry, these 3 components, individually, and then collectively we were able to get about 50% activated substrates. So then we thought this could be good enough. So we added unactivated RNA oligosubstrates to the ribozyme in the presence of these chemicals, and we observed ligation, which means that these unactivated substrates got activated in situ and then got ligated by the ribozymes. So this is preliminary results, which tell us 2 things. One is that RNA-catalyzed RNA ligation is compatible with prebiotic chemistry, and all of this can happen in one pot, which is how things would have happened on early Earth. So the third, again, logical extension to this is that, I just told you that we need enzymes to catalyze RNA ligation efficiently. And then I also told you that these enzymes are ribozymes, which means these enzymes had to be made from RNA itself. So the question is that where do these first ligase ribozyme molecules emerge from? And the logical answer to this has to be through non-enzymatic RNA ligation, right? But the problem here is that this particular reaction, which is non-enzymatic RNA ligation, is a really slow and low-yielding reaction, primarily, I think, because of the low nucleophilicity of the 3' hydroxyl of the RNA. So what we found is that if we modified that 3' hydroxyl with an amino acid group, then that amino group of that amino acid would attack the phosphorus center of the second RNA piece, and this would result in a ligated product where two pieces of RNA would be joined with an amino acid bridge. The good thing about this reaction is it's about 20 to 100-fold faster than an all RNA ligation. So we use this strategy to stitch together pieces of activated amino acid conjugated RNA molecules, and we were able to generate a ligase ribozyme sequence from 4 pieces, as you can see from the gel on the right. Now, mind you, this is not an all RNA ligase ribozyme. This is a chimeric molecule, which is made of the building blocks of both RNA and proteins, right? They contain phosphodiester linkages as well as amino acid bridges in their backbone, which kind of is an interesting thing to think about, because this could represent a conceptual link between the RNA and the protein worlds. But for our purposes, what was really exciting to us is that this was a functional molecule. So this chimeric polymer could actually function as a ribozyme and catalyze the same ligation that I was talking about. And the ligation rates were only about 6-fold slower than an all RNA enzyme. So that was very exciting. So these chimeric ribozymes made of nucleotides and amino acids could sort of point away from the RNA world toward a more modern biology. But another transition that I think would point in the same direction is a transition from these prebiotic RNA building blocks to more modern RNA building blocks. So what I'm talking about here is a transition from 2-amino imidazole activated oligomers to triphosphate activated oligomers. Since ligation is the framework in which I am trying to discuss RNA assembly, I'm going to try to find out evolutionary pathways that could connect ribozymes that ligate AI activated RNA oligomers to those that ligate triphosphate activated oligomers. So to do that, I went back to the ligase ribozymes that I selected at the top of, I discussed at the top of this presentation, and I partially randomized the sequence of these ligase ribozymes, and I repeated our selection, but the only difference here would be, instead of challenging this RNA pool with an imidazole activated substrate, I would challenge it with a triphosphate activated substrate. So after carrying this out for 6 rounds of selection, I obtained an RNA pool that could catalyze the ligation of both AI substrates as well as triphosphate substrates. One thing to note here is that this RNA pool catalyzed the ligation of triphosphate substrates 3,000-fold faster than the corresponding background reaction, which means that catalytic enrichment was happening in this pool. So I sequenced this pool so that I could identify single ribozyme sequences in that pool. And when I did that, it was a surprise to me that the most abundant sequences from this pool were themselves actually promiscuous ribozymes that catalyzed ligation of both triphosphate and amino imidazole activated substrates. So these ribozymes sort of symbolize the evolutionary intermediates in this transition from prebiotic RNA building blocks to modern RNA building blocks. Right, so the last transition that I'm going to talk about is, of course, a very important one, which is the emergence of cellular structures, cell-like compartments where ribozymes could function within them. Many of us are familiar with fatty acid molecules, which are the building blocks of phospholipids. And we think that fatty acids could have been a really attractive candidate for constituting primitive cell membranes. And there are many different reasons mentioned in the literature for this. Some of them are like, fatty acid molecules, at least small fatty acid molecules, can be made by abiotic syntheses. Some of them have also been found in carbonaceous chondrites, which is always exciting. And most importantly, fatty acid molecules self-assemble into spherical vesicles that can encapsulate stuff that's lying around, for example, like RNA. So, RNA enzymes, ribozymes functioning inside fatty acid vesicles appear to be a very exciting model for protocells. But unfortunately, this model has not been explored to its greatest extent, which is, I think, because of the fact that ribozymes need moderate to high concentrations of magnesium to function. But at those concentrations of magnesium, these fatty acid vesicles become leaky and become unstable. So the bottom line here, which is the magnesium problem for cellular life, is that either you have stable compartments or you have active ribozymes, but it's kind of hard to get both working together. So one of the ways in which we solved this problem was to simply identify a single ribozyme sequence that functioned at submillimolar magnesium. So this is a ligase ribozyme that works even at 1 millimolar magnesium, which now provides an excellent opportunity for us to constitute the very first instance of RNA-catalyzed RNA assembly within these prebiotic fatty acid compartments. So this is how these vesicles look under microscopy. They look healthy and handsome after 3 hours in the presence of 3 millimolar magnesium. So this is the experiment that we did. We put our ligase ribozyme, the substrate, and the template within a fatty acid vesicle. And then we added magnesium on the outside to initiate the reaction. The magnesium swims in, catalyzes, starts the reaction. And then we break open these fatty acid vesicles at different time points to analyze its contents. And we're very happy to find that ligation happened robustly within these vesicles. In fact, ligation yields, as you can see, within these vesicles were comparable to ligation observed in solution. So I think this is a small but a necessary step toward our ultimate goal of generating self-replicating protocells where ribozymes make copies of themselves within these prebiotically plausible compartments. This actually has profound implications, right? So imagine you have two protocells with slightly different RNA enzymes in them, right? And then the protocell with a better enzyme will make more RNA in them. So there'll be an RNA buildup, which will make the cells grow fatter and fatter, right? And then they'll make more babies. And these cells will ultimately take over the entire population. And this, of course, is a very special moment because this is where a primitive version of Darwinian evolution would start to emerge spontaneously in systems made with pure chemistry. We are, of course, not here yet, but I hope that meetings such as this and collaborations like this one would ultimately help us to achieve a feat like this in the near future. So yes, that's all I have. So I want to end with that positive note. I hope that we are able to crack this mystery ultimately in the near future. I would like to thank the Shostak Lab and of course Jack and some of the people who were directly involved in the work that I discussed today. And once again, thanks to the organizers and thanks to all of you for listening. I'd be happy to take questions. Thank you.
Thank you very much. So we do have 2 questions. Shashi Tutopali writes, you indeed see a slight increase in the ligation activity within your compartment. Is this simply due to enhanced concentration or is there other interesting physics going on there?
Right. So actually, I don't see an increase in ligation within vesicles. The colors are switched in the bar plot. As you can see from the gel, there is indeed better ligation in solution than inside vesicles. But the black ones here should be in solution and the gray ones should be in vesicles. Can you see the black ones are, they should be in solution and the gray ones should be in vesicles. So that's my mistake. I switched the colors.
Fine. So one last question.
If I could just quickly finish the answer, sorry. So basically, I think that the reason why ligation is slightly slower in vesicles is because the magnesium needs to go from the outside to the inside, and this is why there is less magnesium available to the ribozyme. Sorry, yes, go ahead.
Okay, one more question before the break. You mentioned that a single ribozyme tolerated the magnesium +2. How many ribozymes were not able to tolerate this condition?
We tested about 20 of our sequences that we got out of selection, and none of them worked at concentrations such as 1 millimolar or 2 millimolar. So surprisingly, there was just one sequence that worked at low magnesium.
Okay.
Life is surprising.
Yes.
Yes. But we have figured out more general systems chemistry approaches to reduce the magnesium requirement, which I didn't have time to talk about in today's presentation.
Okay.
Thank you. Wonderful.
Okay, great. Thank you all and meet you soon.